When most of us think about NASA, we imagine rockets leaving Earth. We imagine telescopes aimed at distant stars. We imagine engineers calculating trajectories far beyond our atmosphere. But some of NASA's most advanced equipment does not launch upward. It goes down, miles down, into darkness so complete that sunlight has never touched it. In 1977, a small submersible descended into the Pacific, expecting empty volcanic rock. What it encountered forced scientists to rethink what life requires. That moment did not just change marine biology, it quietly altered the direction of space exploration. Today, NASA astronauts train underwater. Robotic systems designed for distant missions are tested in extreme ocean environments. Sensors built for other worlds are calibrated using data gathered far below the surface of this one. This is not symbolic preparation, it is deliberate. More than 80% of our ocean floor remains unmapped in high detail. Vast regions exist that no human has seen directly. Pressure there exceeds 1,000 times what we experience at sea level. Temperatures swing from near freezing to extremes that would destroy surface machinery. And yet NASA keeps returning, not for spectacle, not for tourism. So why would a space agency repeatedly descend into Earth's deepest waters? What problem can only be studied in total darkness? What is being tested, measured, or prepared beneath miles of ocean? And what does the deep sea reveal that orbit cannot? Tonight, we look beneath the surface of our own planet to understand what NASA may really be searching for. But before we begin, I would love to know where in the world you're listening from and what time it is there. It's always fascinating to think of these stories drifting across so many different skies. If you enjoy these calm journeys into science and mystery, please consider leaving a like and subscribing. It helps this quiet little corner of the internet continue to grow. Now take a slow breath, let your shoulders soften, and let's begin. There are regions of this planet that exist only as gradients on a screen, faint shifts in gravity and contour that hint at mountains no one has seen. The seafloor, vast and folded, remains largely unresolved, its shapes inferred from satellites that measure subtle rises and dips in the ocean's surface far above. More than 80% of it has never been mapped in high detail. In many places, we possess clearer topographic models of Mars than of the terrain beneath our own waters. The red plains of another world have been traced with remarkable precision, while the ground below our oceans lingers in approximation, sketched rather than known. Light does not accompany us very far. Within the first few hundred meters, sunlight thins into a dim blue haze, then dissolves entirely. Below that threshold lies a realm without dawn or horizon, a continuous night that has persisted for millions of years. It is easy to imagine the deep sea as empty, but it forms the largest living space on Earth, an immense volume extending across basins and trenches that wrap around the globe. Most of Earth's biosphere exists in this darkness, sustained not by the steady rhythm of day and night, but by slower exchanges of chemistry and pressure. The scale becomes difficult to hold in the mind. The Mariana Trench descends nearly 36,000 feet, a vertical distance greater than the height of Mount Everest above sea level. At its base rests Challenger Deep, the lowest known point in any ocean. Only a small number of humans have traveled there, enclosed within reinforced spheres designed to withstand forces that would crumple ordinary steel. Outside those hulls, pressure presses inward with a weight that reshapes materials and challenges every seal, every joint, every assumption about durability. Yet even these figures describe only fragments. The deep ocean is not a single pit, but a network of abyssal plains, mid-ocean ridges, subduction zones, and trenches that shift over geological time. Entire mountain ranges rise beneath the waves, their peaks never breaking the surface. Valleys stretch for hundreds of miles, carved by tectonic motion and sediment flow. Hydrothermal systems vent mineral rich fluids into the surrounding water, constructing chimneys that grow slowly in the absence of light. Between these features lie expanses so broad and level that they resemble submerged deserts, though they are anything but barren. What makes this frontier distinct is not only its depth, but its inaccessibility. Weather patterns, surface currents, and storms conceal what lies below. Instruments must be lowered carefully, guided through layers of temperature and density that bend sound and scatter signals. Communication slows, navigation grows uncertain, and each descent becomes an exercise in patience. Data returns in fragments, pieced together over months and years to reveal outlines that remain incomplete. The contrast is quiet but striking. Humanity has placed orbiters around distant planets, landed rovers on alien soil, and charted craters millions of miles away. Meanwhile, the majority of our own seafloor waits in partial shadow, mapped at resolutions too coarse to reveal its true character. This imbalance is not neglect but limitation, a reflection of how difficult it is to explore an environment where pressure climbs relentlessly and light never returns. And so the deep ocean stands as Earth's final frontier, not in rhetoric but in measurable reality. It is a domain that challenges technology, redefines habitability, and humbles certainty. Beneath the waves, the planet holds a record of its history and a laboratory of extremes, extending downward into darkness that remains, even now, only partly understood. The descent began as routine geology. In 1977, the submersible Alvin moved slowly along the Galapagos Rift, its lights cutting across basalt shaped by volcanic forces. The expectation was straightforward, a survey of seafloor spreading, mineral deposits, and the quiet architecture of tectonic motion. No one anticipated a revelation about biology. The cameras were prepared for rock, not for movement. What appeared first was not dramatic in motion, but unmistakable in form. Pale structures rose from the dark seabed, clustered in dense assemblies. They were tube worms, some extending more than 6 feet in length, anchored in chimneys that vented mineral-rich fluids. Around them gathered crabs with white shells, clams embedded in sediment, shrimp drifting in the heated currents. The scene held a kind of steady abundance, an ecosystem arranged around fissures in the crust. It existed where no sunlight had ever reached. The assumption that life required photosynthesis had shaped biological thinking for generations. Energy was believed to flow from the sun, captured by plants, passed upward through grazing and predation. Here, that sequence was absent. The foundation of this community was chemical, not solar. Microorganisms within the vent fields oxidized hydrogen sulfide and other compounds released from beneath the crust. Through chemosynthesis, they converted chemical gradients into usable energy, forming the base of a food web independent of daylight. The tube worms themselves carried no mouths or digestive tracts. Inside their tissues lived symbiotic bacteria that performed this chemistry on their behalf. Hemoglobin in the worm's blood bound hydrogen sulfide without poisoning the host, transporting it to the microbes within. The relationship was precise, evolved over deep time, tuned to conditions once thought inhospitable. Around them, other organisms adapted to fluctuating temperatures and shifting mineral flows, forming a stable system in a place defined by extremes. This finding did more than expand a catalog of species. It altered the conceptual boundaries of habitability. If life could organize itself around chemical energy drawn from planetary interiors, then sunlight was no longer the sole architect of ecosystems. The implications extended backward toward early Earth, where similar hydrothermal environments may have existed long before the atmosphere stabilized. They also extended outward toward icy moons and distant worlds. Within NASA's astrobiology community, the connection was immediate. Models of Europa, with its subsurface ocean warmed by tidal flexing, gained new relevance. Enceladus, venting water vapor and organic molecules into space, began to resemble not an abstract curiosity, but a potential analog. Even bodies far from the sun, once dismissed as too cold and too dim, entered scientific conversation with renewed seriousness. The discovery at the Galapagos Rift provided a working example that life need not rely on surface light if internal chemistry could supply energy. The Alvin expedition did not announce a revolution in dramatic tones. It returned samples, images, measurements. Papers were written, hypotheses refined, textbooks reconsidered. Over time, hydrothermal vent systems were found in other oceans, confirming that this was not an isolated anomaly, but a recurring feature of Earth's geology. Each new site reinforced the same principle that biological systems can anchor themselves to gradients of heat and chemistry rising from below. In that steady accumulation of evidence, a quiet shift occurred. The map of where life might exist expanded, not through speculation, but through observation. A volcanic rift beneath the Pacific had offered a new template, one that would guide instruments, missions, and expectations far beyond Earth's horizon. Energy, in its most familiar form, arrives from above. It spills across continents, warms oceans, and fuels the green surface of the world. Yet far below that illuminated skin, another economy operates, slower and more concealed, drawing its strength from gradients hidden within rock and water. In the depths surrounding hydrothermal systems, life organizes itself around chemistry, not light, and the foundation of that organization is chemosynthesis. At the boundary where heated fluids meet cold seawater, dissolved compounds stream outward from the crust. Among them is hydrogen sulfide, a molecule toxic to many surface organisms, yet rich in potential energy. Specialized microbes cluster along these mixing zones, their cellular machinery tuned to exploit chemical disequilibrium. Through metabolic pathways that couple oxidation and reduction reactions, they transform hydrogen sulfide into usable biochemical energy. Carbon dioxide becomes organic matter, not through sunlight driven photosystems, but through enzymes that harvest electrons from mineral rich fluids. The result is a primary production system anchored entirely in geochemistry. From this microbial base, larger organisms assemble. Symbiotic relationships form with precision. In some species, internal tissues house dense colonies of bacteria, each partner dependent on the other. The host circulates sulfide and oxygen through its body, while the microbes synthesize nutrients that sustain growth. Elsewhere, grazing species feed directly on bacterial mats coating mineral surfaces. Predators move among them, linking the tiers of this chemically powered web. No photons are required. Instead, the gradient between hot vent, effluent, and surrounding seawater becomes the engine of continuity. This realization extended beyond ecology into the deeper logic of life's requirements. Isolation from sunlight did not equate to sterility. As long as a planet maintains internal heat, circulates water, and sustains chemical contrasts, metabolic pathways can emerge. The essential ingredient is not brightness but imbalance, a flow of electrons from one state to another. In these submerged systems, rock interacts with water to create that imbalance, sustaining communities in places once dismissed as inhospitable. Such environments offer a window into the distant past. Early Earth lacked the stable oxygen rich atmosphere known today. Volcanism was widespread. Oceans interacted vigorously with newly formed crust, and chemical gradients were abundant. Some origin of life models propose that mineral surfaces near hydrothermal systems acted as scaffolds for complex chemistry. Iron sulfur minerals, common in vent settings, can catalyze reactions that assemble organic molecules from simpler precursors. Within microscopic pores of mineral structures, temperature and chemistry fluctuate in patterns that may have encouraged self organizing processes. In that sense, modern vent ecosystems may echo conditions that once shaped the first replicating systems. The relevance of these ideas extends outward into planetary science. Worlds far from the sun may still harbor internal heat generated by radioactive decay or tidal flexing. If water exists beneath ice shells, and if rock interacts with that water, chemical energy could accumulate in similar ways. The lesson drawn from Earth's vents is not speculative optimism, but empirical precedent. Biology has demonstrated that it can anchor itself to planetary interiors, provided certain physical and chemical thresholds are met. Within laboratories and research vessels, these insights translate into measurable criteria. Scientists examine redox couples, mineral assemblages, and the stability of organic compounds under pressure and temperature extremes. Instruments are designed to detect not only sunlight derived signatures, but also byproducts of chemical metabolism. The concept of habitability broadens quietly, grounded in observations from Earth's own depths. In the steady flow of sulfide rich fluids and the patient work of microbes, a different blueprint for living systems takes form, one that reshapes how environments are evaluated, both here and elsewhere. In the absence of familiar reference points, anatomy begins to take on unfamiliar shapes. Forms emerge that seem assembled from a different blueprint, as though evolution, given enough isolation and time, has rewritten its own constraints. Along vent fields and in abyssal basins, organisms persist whose structures would appear improbable in shallower seas, yet here they function with steady precision. Consider the tall, pale tube worms anchored to mineral formations. They possess no mouths, no stomachs, no digestive tracts in the conventional sense. Their survival depends entirely on dense colonies of bacteria housed within a specialized internal organ. Blood rich in hemoglobin circulates through their bodies, binding both oxygen and hydrogen sulfide, delivering these molecules to symbiotic microbes that synthesize organic compounds. The worm provides transport and protection. The bacteria provide nourishment. It is a closed partnership, self-contained and efficient, sustained by chemistry rising from the crust. Elsewhere, deep-sea shrimp drift through heated plumes where visibility remains limited. On their backs are light-detecting organs, positioned not to scan the sky, but to register faint emissions from vent systems. These photoreceptors allow them to orient toward gradients of warmth and chemical flux, a sensory arrangement inverted from surface expectations. Vision becomes a tool not for sunlight, but for the subtle glow of mineral reactions in water. Fish adapted to polar and abyssal conditions circulate antifreeze proteins within their bloodstreams. These molecules bind to forming ice crystals, inhibiting their growth and preventing cellular damage in near freezing water. The protein structures align with crystal faces, interrupting expansion at the microscopic scale. In doing so, they preserve fluid circulation in temperatures that would immobilize unprotected tissues. The adaptation is molecular, invisible without instrumentation, yet essential for survival in cold, stable depths. Microbial communities extend tolerance even further. Certain thermophilic species thrive at temperatures exceeding the boiling point of water at surface pressure, stabilized by the immense forces of the deep. Their enzymes remain folded and functional under heat that would denature most proteins. Membrane lipids form tightly packed layers, reducing permeability and maintaining integrity. In contrast, piezophilic microbes endure crushing pressure that would deform metal hulls. Their cellular components adjust to compression, proteins shaped to operate efficiently when squeezed into configurations intolerable to other life forms. DNA repair systems counteract damage induced by high pressure environments, sustaining replication over generations. These adaptations do not occur in isolation. They assemble into communities structured around gradients of temperature, chemistry, and pressure. Predators, grazers, symbionts, and scavengers occupy defined roles, forming networks that persist across years and decades. Population dynamics fluctuate with geological activity, yet the underlying systems remain coherent. What might appear extraordinary from a surface perspective functions as ordinary within this context. The cumulative effect of these organisms is not spectacle, but recalibration. The boundaries once assumed to define habitability shift quietly outward. Biological resilience expresses itself not as defiance, but as accommodation, each species tuned to a narrow band of environmental variables. Stability arises from specialization, from precise biochemical pathways and structural modifications that align with local conditions. Within these ecosystems, life does not merely endure extremes, it integrates them into routine existence. Pressure becomes background, heat a resource, chemical toxicity a substrate for metabolism. The result is not a collection of anomalies, but a sustained biosphere, operating beyond prior expectations. In observing these systems, a broader pattern becomes visible, one in which adaptability is not marginal, but central to life's continuity across the planet's varied domains. Beneath the shifting sediments and fractured basalt of the seafloor lies another realm that does not announce itself through visible structures or drifting bodies. It is not arranged around chimneys or currents, but embedded within the rock itself. Pores the width of a human hair, microscopic cracks formed by tectonic stress, and mineral boundaries, where water seeps slowly through stone, become shelters for organisms too small to see without magnification. Kilometers below the ocean floor, microbial life persists in darkness so complete that even the faint glow of vent systems never reaches. These microorganisms do not float freely. They inhabit the interstices of basalt, sandstone, and ultramafic rock, forming thin biofilms along mineral surfaces. Water percolates downward through permeable layers, carrying dissolved compounds that react with surrounding material. In these confined spaces, chemical reactions unfold between water and iron-rich minerals, releasing hydrogen and altering oxidation states. Microbes capitalize on these gradients, drawing energy from redox reactions that require no connection to the sunlit world above. Their metabolism proceeds slowly, measured in years rather than hours, yet it continues with steady persistence. The environment is austere, nutrient flux is limited, temperatures vary depending on depth and geothermal flow, and pressure remains immense. Yet these cells maintain membranes, replicate DNA, and repair molecular damage. Some species divide only once in decades, conserving energy with extraordinary efficiency. Their enzymes are adapted to function under compression and elevated heat, maintaining structure where other proteins would unravel. Over geological time, populations establish equilibrium with their surroundings, neither expanding rapidly nor vanishing, but existing in long-term balance with the chemistry of stone. Sampling this hidden biosphere requires careful drilling and sterile technique. Core samples, retrieved from beneath the seabed, reveal living cells in sediments millions of years old. In certain regions, microbial communities extend several kilometers downward, following fractures created by plate movement. The cumulative biomass of these subsurface populations may represent a substantial fraction of Earth's total microbial life. Estimates suggest that a significant portion of the planet's prokaryotes reside not in forests or oceans, but within crustal rock and buried sediment. Isolation defines this domain. Surface ecosystems cycle nutrients through sunlight driven processes, exchanging gases with the atmosphere and responding to seasonal rhythms. The deep biosphere operates independently, disconnected from weather and photosynthesis. Energy arises from interactions between water and minerals, not from solar radiation. Carbon moves through slow subterranean pathways, and metabolic waste diffuses gradually through pore networks. Entire communities persist without ever encountering the surface, their lineage unfolding in concealed corridors beneath the ocean floor. This discovery has reshaped scientific understanding of habitability. The presence of liquid water, reactive minerals, and internal heat becomes sufficient for sustaining life, even in the absence of surface illumination. Planets and moons once deemed sterile due to lack of sunlight now appear differently when viewed through this lens. If subsurface oceans or hydrated crusts exist elsewhere, and if chemical gradients are present, microbial systems could emerge and endure beyond observation. The deep biosphere does not present dramatic imagery. It offers something subtler, a recalibration of scale. Life is not confined to surfaces or illuminated zones. It threads through rock, inhabits microscopic voids, and persists in environments once considered inert. Within the quiet architecture of the crust, metabolism continues, redefining the boundaries of where living systems can reside and expanding the map of habitable space beneath our own feet. Far beyond the orbit of Mars, where sunlight thins and the sky darkens into a deeper shade of black, a small moon circles Jupiter in quiet persistence. Europa appears at first as a pale sphere, its surface etched with long fractures and intersecting khatut of reddish-brown material. Beneath that frozen exterior, evidence suggests the presence of a vast ocean concealed under kilometers of ice. This hidden reservoir may contain more liquid water than all of Earth's oceans combined, held not in open waves but in darkness beneath a rigid shell. The source of warmth within Europa does not come primarily from the distant Sunday. Instead, gravity performs the work. As Europa travels along its slightly elliptical orbit, Jupiter's immense pull stretches and compresses the moon in a rhythmic cycle. These tidal forces generate internal friction, heating the subsurface layers through mechanical stress. Ice shifts, cracks, and refreezes at the surface, while deeper regions may remain liquid under sustained pressure and warmth. In this way, energy circulates from orbital motion into thermal gradients within the moon's interior. Models of Europa's structure describe a layered world, an outer crust of water ice, fractured and mobile, overlays an ocean that may extend tens to perhaps 100 kilometers in depth. Beneath that ocean lies a rocky mantle, potentially interacting with the water above. If heat from tidal flexing penetrates downward, it could drive chemical reactions at the boundary between rock and ocean. On Earth, such interactions support complex microbial systems. On Europa, similar processes remain hypothetical, yet they align with the physics of water and mineral contact under pressure. Observations from past spacecraft, including data gathered during flybys of the Jovian system, have revealed surface features that hint at subsurface movement. Chaotic terrains appear where ice has broken apart and reassembled, suggesting exchange between the ocean below and the crust above. Spectroscopic measurements detect salts and possible organic compounds embedded in surface materials. These clues do not confirm biology, but they indicate a dynamic environment where chemistry is active rather than static. Hydrothermal activity on Europa's seafloor remains an open possibility. If tidal heating extends into the rocky interior, fractures could allow seawater to interact with warm mineral surfaces. Such reactions may produce hydrogen and other reduced compounds capable of fueling metabolic processes. The parallels to Earth's deep systems are not direct replicas, but they offer a conceptual framework for evaluating habitability beyond sunlight. In this context, the essential criteria become liquid water, sustained energy, and chemical diversity. To explore these possibilities, NASA has developed the Europa Clipper mission. Rather than landing immediately, the spacecraft is designed to perform repeated close flybys, mapping the moon's surface and analyzing its composition with refined instruments. Radar systems will probe the thickness of the ice shell, while spectrometers examine surface chemistry. Magnetometers will measure variations in Europa's magnetic field, providing indirect evidence about the depth and salinity of the ocean beneath. Each dataset contributes to a layered understanding of this concealed world. Europa stands as a reminder that oceans need not be illuminated to exist. Encased in ice and distant from the sun's warmth, it may still sustain internal cycles of energy and chemistry. The concept of an ocean world expands from Earth's seafloor into the outer solar system, suggesting that habitable environments may arise in places once considered too cold and remote. In the steady orbit of this moon, gravitational forces shape a hidden sea, inviting careful observation and measured exploration from afar. Against the pale rings of Saturn, a small moon moves with quiet regularity, its surface bright as fresh snow. Enceladus appears modest in size, yet from fractures near its southern pole, narrow jets of water vapor stream outward into space. These plumes rise hundreds of kilometers above the surface, forming a faint halo that feeds Saturn's E-ring. What escapes through those cracks does not disperse as empty mist. It carries within it a chemical record of the ocean concealed below. Data gathered by the Cassini spacecraft transformed Enceladus from a distant curiosity into a world of active processes. As Cassini passed repeatedly through the plumes, its instruments sampled microscopic ice grains and vapor. Analysis revealed the presence of organic molecules embedded within those particles, carbon-containing compounds that form the structural backbone of living systems on Earth. These organics were not arranged into complex organisms, yet their detection signaled that Enceladus hosts chemistry more intricate than frozen water alone. Among the most significant findings was molecular hydrogen. In terrestrial environments, hydrogen often emerges where water interacts with hot rock, particularly through processes such as serpentinization. When liquid water permeates iron-rich minerals under heat and pressure, chemical reactions generate hydrogen gas along with altered mineral phases. On Earth, microbial communities exploit this hydrogen as an energy source, coupling it with carbon dioxide to drive metabolism. The presence of hydrogen within Enceladus's plumes suggests that similar water-rock interactions may be occurring beneath its icy shell. Models of Enceladus propose a subsurface ocean resting atop a rocky core. Tidal forces exerted by Saturn flex the moon's interior, producing frictional heating sufficient to maintain liquid water. If that ocean comes into contact with the rocky core, hydrothermal circulation becomes plausible. Warm water rising through fractures could leach minerals, transport dissolved compounds, and eventually vent material upward through surface fissures. The plumes then act as natural conduits, delivering subsurface samples directly into space, accessible to passing spacecraft. The chemical composition measured by Cassini aligns with this scenario. In addition to hydrogen and organics, the plume particles contained salts and silica grains, the latter indicative of high temperature interactions between water and rock. Laboratory simulations demonstrate that silica nanoparticles can form under hydrothermal conditions, reinforcing the interpretation that Enceladus may harbor active seafloor processes. The parallels to Earth's deep marine systems are not superficial comparisons. They arise from shared physical and chemical principles governing fluid circulation and mineral exchange. Insights from oceanographic research provide context for evaluating these extraterrestrial measurements. Studies of vent chemistry, redox gradients, and microbial energetics inform the thresholds at which metabolism becomes feasible. By understanding how hydrogen and carbon compounds sustain communities in Earth's depths, scientists refine models for potential ecosystems on Enceladus. The interpretation remains cautious, grounded in chemistry rather than assumption. Yet the alignment between plume composition and known hydrothermal reactions narrows the gap between speculation and plausibility. Enceladus presents an unusual circumstance in planetary exploration. Its ocean, though hidden, announces itself through continuous ejection. Instead of drilling through kilometers of ice, a spacecraft can traverse the plumes and analyze their contents directly. Each pass through the vapor cloud gathers additional data, incrementally clarifying the structure and chemistry of the subsurface sea. Within those frozen grains and drifting molecules lies evidence of internal activity, shaped by tidal energy and water-rock contact, echoing processes observed in Earth's own hidden oceans. A dense orange haze shrouds Saturn's largest moon, softening its outline and concealing the surface beneath a layered atmosphere. Titan moves through space with a quiet steadiness, its skies thick with nitrogen and hydrocarbons that scatter sunlight into a muted glow. Radar instruments have pierced that haze to reveal shorelines, channels, and broad basins filled not with water, but with liquid methane and ethane. These lakes rest at temperatures far below freezing by Earth standards, yet they remain fluid under Titan's frigid conditions, forming a hydrological cycle built from entirely different materials. Clouds condense and drift across Titan's skies, releasing methane rain that carves branching river networks into the landscape. Dunes of dark organic particles stretch across equatorial regions, shaped by winds moving through the dense air. The chemistry of this world unfolds slowly, driven by ultraviolet radiation from the sun and energetic particles trapped within Saturn's magnetic field. These interactions break apart simple molecules in the upper atmosphere, allowing them to recombine into more complex hydrocarbons and nitriles that settle gradually toward the surface. The resulting environment is rich in organic compounds, though not in liquid water at the surface. Laboratory simulations reproduce Titan's atmospheric chemistry, yielding tholins, complex macromolecules that resemble the building blocks of prebiotic systems. While the temperature remains too low for familiar aqueous biology to function, the abundance of carbon-based chemistry broadens the scope of possible biochemical frameworks. Some researchers explore whether alternative solvents, such as liquid methane or ethane, could support different forms of molecular organization. The constraints are severe. reaction rates slow, and stability requirements stringent, yet the theoretical space expands beyond traditional models. Insights from Earth's cold seep ecosystems provide a grounded comparison. In certain marine environments, methane escapes from sediments and interacts with surrounding seawater, forming localized chemical gradients. Microbial communities exploit these gradients, oxidizing methane and coupling it with sulfate reduction to generate energy. Though Titan's conditions differ markedly in temperature and composition, the principle that metabolism can arise from chemical disequilibrium rather than sunlight remains relevant. Studying such Earth systems refines understanding of how non-solar energy pathways might operate under constrained conditions. Titan's subsurface may also conceal water, locked beneath layers of ice and hydrocarbon-rich crust. Models suggest the presence of an internal ocean composed of water mixed with ammonia, maintained in liquid form by residual heat. If such an ocean exists, interactions between water and rock could produce chemical gradients similar to those inferred on other icy worlds. In that case, Titan would contain two distinct regimes: a hydrocarbon surface cycle and a deeper aqueous environment, each governed by different physical laws, yet connected within a single planetary system. The significance of Titan lies not in confirmed biology, but in conceptual extension. It challenges assumptions about the range of environments where complex chemistry can persist. Liquid methane lakes, hydrocarbon rain, and atmospheric synthesis of organic molecules illustrate that planetary processes can generate intricate chemical landscapes far from the warmth of a star. These landscapes invite careful modeling and experimental replication, allowing scientists to test the boundaries of molecular stability and potential metabolism. Through Titan, the definition of habitability becomes more elastic. The requirement shifts from Earth-like conditions to the broader presence of liquid phases, energy gradients, and chemical diversity. Even in an environment defined by cold and haze, processes unfold that echo fundamental principles observed elsewhere. Within that orange shroud, chemistry advances patiently, expanding the map of possible living systems and deepening the dialogue between planetary science and biology. In conference rooms far from launch pads and clean rooms, mission priorities are shaped by a simple shift in perspective. The search for life is no longer confined to rocky planets bathed in starlight. Within NASA's strategic planning, a category has taken formal shape under the name Ocean Worlds, a designation that recognizes the scientific weight of planets and moons believed to harbor liquid reservoirs beneath their surfaces. This framework brings together bodies such as Europa, Enceladus, and Titan, not as isolated curiosities, but as members of a shared class defined by internal oceans and sustained energy. The emphasis on these worlds reflects an accumulation of evidence gathered both in orbit and beneath Earth's own waters. Data from submersible expeditions, drilling programs, and chemical surveys have clarified the conditions under which life persists without sunlight. Those findings now inform the criteria used to evaluate distant environments. When engineers outline mission objectives, they incorporate parameters refined through oceanographic research, including temperature ranges, pressure tolerances, and chemical signatures associated with metabolic processes. Planning for exploration of icy moons demands careful integration of planetary science and marine biology. If a spacecraft is to investigate the chemistry of a subsurface ocean, it must be equipped to detect dissolved gases, organic compounds, and mineral interactions analogous to those studied on Earth. Instruments capable of identifying trace concentrations of hydrogen, methane, or complex carbon molecules are calibrated using samples and datasets derived from terrestrial expeditions. Spectrometers, mass analyzers, and chromatographs are tested against standards informed by deep ocean chemistry, ensuring that sensitivity aligns with realistic expectations. Beyond chemical detection, structural design also draws from underwater experience. Autonomous underwater vehicles navigating Earth's abyss have demonstrated how machines operate in environments where communication delays, pressure, and darkness impose strict limits. Lessons learned in stabilizing sensors, protecting electronics from compression, and managing limited power budgets translate into spacecraft systems intended for distant ocean worlds. The engineering does not replicate marine vehicles directly, yet the constraints overlap sufficiently to provide valuable guidance. The Ocean Worlds program also shapes decisions about planetary protection. Preventing contamination of potentially habitable environments requires sterile assembly procedures and containment strategies developed through years of drilling and sampling in extreme settings. Techniques refined during studies of subsurface ecosystems, where even minor biological intrusion can distort results, contribute to protocols governing spacecraft destined for icy moons. The objective is preservation of scientific integrity, allowing future measurements to reflect indigenous chemistry rather than imported biology. Strategic sequencing of missions reflects this evolving framework. Rather than pursuing immediate landings, NASA often begins with orbital reconnaissance and repeated flybys, building layered models of surface composition and internal structure. Gravity measurements, magnetic field analyses, and ice-penetrating radar surveys create a foundation upon which more ambitious exploration may later rest. Each phase incorporates insights gained from Earth's oceans, where incremental mapping and sampling have proven essential for understanding complex systems. The designation Ocean Worlds represents more than a thematic label. It signals an institutional acknowledgement that liquid water, internal heat, and chemical gradients can coexist far from the traditional habitable zone. By aligning planetary missions with knowledge drawn from Earth's hidden seas, NASA integrates disciplines that once operated separately. The ocean beneath our own planet becomes a training ground in both concept and technique, shaping how instruments are built, how data are interpreted, and how distant oceans are approached with measured curiosity and technical precision. Long before a spacecraft ever leaves Earth, its technologies often descend into water. In harbors, research basins, and open ocean test ranges, autonomous underwater vehicles slip beneath the surface, carrying experimental navigation systems and sensor arrays. These machines move without direct tether, guided by algorithms that must interpret incomplete data and adjust to shifting conditions. In the absence of GPS signals and constant communication, they rely on inertial measurement units, sonar mapping, and onboard decision-making to maintain course. Each dive becomes a rehearsal in autonomy, a controlled study of how a machine behaves when distance and delay separate it from human oversight. Water offers a particular kind of challenge. Signals scatter, visibility diminishes, and currents introduce subtle drift. Engineers observe how navigation software compensates for these uncertainties, refining code that may one day guide probes across alien terrain. When an underwater vehicle must identify obstacles or map an unfamiliar landscape, it constructs a model from echoes and gradients rather than direct sight. This reliance on indirect perception parallels the constraints expected for missions to distant ocean worlds, where communication delays will require spacecraft to interpret and respond without immediate instruction. In polar regions, The rehearsal grows more specific. Beneath Antarctic ice shelves, ice-penetrating probes are lowered through drilled boreholes into subglacial waters. These environments simulate key aspects of icy moons, including isolation from sunlight, limited access points, and the need for sterile operation. Engineers test drilling techniques that prevent contamination, monitor how instruments function in near freezing conditions, and evaluate the durability of seals and electronics exposed to prolonged cold. The Antarctic setting does not replicate extraterrestrial oceans exactly, yet it provides a realistic analog for the engineering problems that must be solved. Pressure testing is equally essential. Deep ocean environments subject housings and connectors to forces that compress materials and stress joints. By deploying equipment at significant depth, designers learn how components deform, how cables transmit data under load, and how insulation behaves over extended periods. Failure points discovered underwater inform revisions before any hardware is integrated into spacecraft. The ocean becomes a proving ground where weaknesses can be identified and corrected within reach of recovery teams. Darkness adds another dimension. Optical systems calibrated for low light conditions are evaluated in turbid waters where illumination is minimal. Cameras, laser scanners, and chemical sensors are assessed for sensitivity and noise reduction. Data collected in these settings allow engineers to adjust thresholds and filtering techniques, ensuring that faint signals are distinguishable from background interference. The goal is not to replicate alien oceans in full detail, but to expose instruments to comparable constraints and observe their response. Through repeated deployments, patterns emerge. Autonomous control improves, power management becomes more efficient, and hardware designs converge toward robustness. The lessons are cumulative, each field test contributing to a broader understanding of how machines perform when removed from constant supervision. Underwater trials compress years of theoretical planning into tangible experience, revealing interactions between software, materials, and environment that simulations alone cannot capture. In this way, robots dive before they fly. The descent into Earth's waters shapes the ascent into space, linking marine engineering with planetary exploration. What is learned in the pressure and cold of terrestrial oceans becomes embedded in spacecraft architecture, guiding how probes will navigate, sample, and endure when they reach distant seas concealed beneath ice. Inside a vast enclosed pool in Houston, spaceflight begins without a rocket. The water is clear, temperature controlled, and deep enough to submerge full-scale replicas of spacecraft components. Suspended within it, suited astronauts move slowly along truss structures and mock airlocks, their motions deliberate and measured. The neutral buoyancy laboratory does not imitate the vacuum of space, yet by carefully adjusting ballast and suit weight, engineers achieve neutral buoyancy, allowing bodies to float with neither sinking nor rising. In that suspended state, the sensation approximates microgravity, providing a practical rehearsal for work beyond Earth. Every movement underwater demands patience. Tools must be secured, tethers managed, and orientation maintained without the steady pull of gravity. Divers positioned nearby observe and assist, while surface teams monitor communication channels and physiological data. The resistance of water differs from the emptiness of orbit, yet it introduces its own discipline. Astronauts learn to plan sequences of motion, to anticipate inertia, and to conserve energy during extended tasks. Hours spent beneath the surface translate into confidence during real spacewalks, where mistakes carry greater consequence. The laboratory is only one aspect of underwater preparation. Off the coast of Florida, And in other marine locations, NASA conducts NEMO missions, short for NASA Extreme Environment Mission Operations. Crews inhabit an underwater research station, living in confined quarters for days or weeks at a time. Outside the habitat, they conduct simulated extravehicular activities on the seafloor, communicating with mission control under structured delays. Inside, they manage daily routines within limited space, sharing resources and responsibilities. Life within an underwater habitat requires adaptation. Sound carries differently, and the presence of surrounding water shapes every excursion. Psychological dynamics become as important as technical skill. Isolation from the surface, even at relatively shallow depths, creates a contained environment where teamwork and clear communication determine success. Schedules are carefully structured, yet flexibility remains essential when unforeseen challenges arise. The experience mirrors aspects of long-duration space missions, where crews must rely on each other while separated from immediate support. Medical and behavioral researchers observe these missions closely. They study how individuals respond to confinement, how decision-making evolves under delayed communication, and how group cohesion is maintained over time. Physiological changes are monitored, including sleep patterns and stress markers. These observations inform planning for future expeditions beyond low Earth orbit, where autonomy will be critical and resupply infrequent. The underwater setting provides a manageable analog, allowing adjustments to protocols before greater distances are attempted. Training beneath the surface also reinforces procedural discipline. Checklists are rehearsed, emergency responses practiced, and contingency plans tested in a medium that demands coordination. Water, though forgiving compared to vacuum, still imposes constraints that reveal weaknesses in preparation. Equipment failures can be simulated, requiring crews to adapt within a structured framework. Each scenario refines habits that will later be applied in orbit or deep space. Through these submerged exercises, astronauts cultivate familiarity with environments that resist ordinary movement and perception. The pool in Houston and the habitats offshore become transitional spaces, bridging Earth and orbit. In their quiet immersion, technical skill and human resilience are shaped together, preparing crews for journeys that extend far above the waterline yet are rehearsed in its depths. Time stretches differently when distance intervenes. In certain underwater missions, a message sent from the surface does not arrive instantly. Instead, it is routed through structured delays, introduced deliberately to mirror the lag that would occur between Earth and a distant spacecraft. Voices that might otherwise overlap in quick exchange are separated by measured silence. Instructions pause in transit. Responses return moments later, shaped by the interval. Beneath the surface, crews conducting simulated exploration adjust to this altered rhythm. Communication links remain active, yet immediacy is removed. A query posed from mission control cannot be clarified in the next breath. Technical anomalies, routine decisions, even small uncertainties must be addressed locally before guidance can be received. The environment is controlled, the risks managed, but the psychological texture shifts. Autonomy becomes more than a procedural term. It becomes the default mode of operation. The rationale is grounded in orbital mechanics. When spacecraft travel toward Mars, radio signals require several minutes to traverse the distance, depending on planetary alignment. During periods of maximum separation, a message may take more than 20 minutes one way. A simple exchange can span nearly an hour. In that interval, crews on a planetary surface cannot rely on immediate confirmation or correction from Earth. Decisions must be made with the information at hand, supported by training and predefined protocols. Underwater analog missions recreate this constraint within a terrestrial setting. Schedules are organized so that mission control communicates through delayed channels. Crew members log observations, execute planned tasks, and respond to simulated malfunctions without expecting instant feedback. The imposed latency reshapes group dynamics. Leadership roles become clearer as individuals assume responsibility for resolving issues internally. Discussion among team members deepens, since consensus must be reached before transmitting conclusions outward. Researchers observe these exercises with attention to subtle details. They examine how stress levels fluctuate when communication slows, how cognitive workload changes, and how trust within the team evolves. Behavioral scientists analyze conversation patterns, noting shifts in tone and brevity when responses are deferred. The data inform planning for missions that may extend for months or years, where prolonged separation from Earth will be unavoidable. Isolation compounds the effect of delay. In underwater habitats, crews inhabit confined quarters removed from ordinary routines. External stimuli are limited, and the surrounding environment reinforces the sense of separation. Within this contained setting, delayed communication highlights the psychological distance from support systems. Yet it also reveals adaptability. Over time, participants often report increased confidence in their own judgment, greater cohesion among teammates, and a more deliberate pace of problem solving. Technical systems are evaluated alongside human performance. Communication protocols are tested for clarity, ensuring that instructions remain unambiguous even when feedback is postponed. Data management practices are refined so that information can be archived, transmitted, and reviewed efficiently across temporal gaps. These operational adjustments contribute to a broader framework for long-duration exploration, where autonomy and reliability must coexist. Through these measured delays, a rehearsal unfolds for journeys far beyond Earth. The underwater setting offers a manageable space in which to explore the implications of distance without leaving the planet. In the quiet intervals between transmitted words, crews learn to navigate not only physical tasks, but also the subtle psychology of separation, preparing for missions where patience and self-reliance will define daily life. Cleanliness, in the context of planetary exploration, becomes an ethical boundary as much as a technical requirement. When a spacecraft departs Earth carrying instruments designed to search for life, it must not carry unintended passengers. Microorganisms are resilient, persistent, and often invisible without magnification. To preserve the integrity of distant environments, NASA applies rigorous planetary protection standards, ensuring that the search for life does not inadvertently transport it. The concern is not abstract. On Earth, microbes inhabit surfaces, air, soil, and even the interiors of carefully maintained facilities. Many species endure desiccation, radiation, and temperature extremes. Some form spores capable of surviving for extended periods under harsh conditions. If such organisms were to reach a moon with subsurface water or a planet with transient liquid phases, they could compromise scientific results. A single contaminant might multiply, obscuring the chemical signatures that missions are designed to interpret. To address this challenge, engineers and microbiologists collaborate in environments that resemble surgical theaters more than industrial workshops. Spacecraft components are assembled in clean rooms where air is filtered, humidity controlled, and particulate counts continuously monitored. Surfaces are sterilized using heat, chemical treatments, or ultraviolet radiation, depending on material constraints. Biological assays quantify the number of viable organisms remaining on hardware, ensuring compliance with strict thresholds established through international agreements. Deep drilling projects on Earth provide an additional layer of expertise. When researchers seek to sample subsurface ecosystems beneath the ocean floor or within polar ice, they confront similar concerns about contamination. A drill string descending kilometers into sediment must not introduce surface microbes into ancient strata. Sterile drilling fluids, sealed core liners, and tracer compounds are employed to detect and minimize intrusion. These techniques, refined through years of oceanographic research, inform protocols for accessing potential habitats on other worlds. Ocean drilling research also advances clean technology relevant to spacecraft design. Instruments intended to penetrate icy crusts or analyze plume material must maintain internal sterility throughout launch, transit, and operation. Lessons learned from handling delicate marine samples translate into improved containment systems, specialized seals, and contamination monitoring procedures. Engineers adapt these methods to withstand vacuum, radiation, and extreme cold, balancing sterility with durability. Planetary protection operates in two directions. In addition to safeguarding other worlds from terrestrial microbes, missions must prevent potential extraterrestrial materials from posing risks upon return. Sample return capsules are designed with layered containment, ensuring that collected material remains isolated during transit through Earth's atmosphere. Facilities prepared to receive such samples are equipped with redundant filtration and isolation systems, drawing from biosafety practices developed in medical and environmental laboratories. Underlying these measures is a principle of scientific responsibility. The objective is not merely to comply with regulation, but to preserve the authenticity of discovery. If signs of biological activity are detected on another world, researchers must be confident that those signals are indigenous rather than imported. The credibility of interpretation depends on meticulous preparation long before a launch window opens. Through sterile assembly, controlled drilling, and disciplined protocol, planetary protection becomes an extension of exploration itself. It recognizes that curiosity must be matched with restraint, and that the act of searching carries consequences. By refining methods on Earth's oceans and subsurface environments, NASA strengthens its capacity to approach distant habitats with care, ensuring that the quest to understand life beyond our planet does not alter the very systems it seeks to study. From orbit, the ocean appears smooth, a broad expanse reflecting sunlight in shifting patterns of blue and silver, yet its surface is not perfectly level. Subtle bulges and depressions ripple across it, differences measured not in meters but in centimeters. These variations are shaped by gravity itself, responding to the mass of structures hidden far below. Satellites circling Earth record these minute changes in sea surface height with extraordinary precision, transforming faint distortions into maps of the unseen. Where a submerged mountain rises from the seafloor, its mass exerts a slightly stronger gravitational pull. Water above that feature gathers imperceptibly, forming a gentle mound detectable only through radar altimetry. Conversely, deep trenches and lighter crustal regions create slight depressions in the ocean surface. By tracking these contours across vast distances, scientists reconstruct the outlines of underwater ridges, fracture zones, and basins that have never been directly surveyed. The technique relies on repeated passes of satellites equipped with altimeters capable of measuring distance to the sea surface within centimeters. Each pulse of radar reflects off the water and returns to the spacecraft, allowing calculation of surface height relative to a reference ellipsoid. Over time, millions of measurements accumulate, building a global dataset that reveals gravitational anomalies. These anomalies correspond to variations in the distribution of mass within Earth's crust and mantle, including features buried beneath kilometers of water. Gravity mapping extends beyond simple visualization. It provides insight into tectonic processes shaping the ocean floor. Mid-ocean ridges, where plates diverge and new crust forms, create distinct gravitational signatures. Subduction zones, where one plate slides beneath another, produce elongated depressions and complex mass distributions. Transform faults and seamount chains leave linear traces across the data. Through careful interpretation, researchers infer the architecture of regions too remote or deep for routine ship-based surveys. This orbital perspective complements direct exploration rather than replacing it. Sonar mapping conducted by research vessels yields high-resolution images of specific areas, but ships can traverse only a fraction of the global ocean. Satellite altimetry, in contrast, offers near-continuous coverage, identifying areas of interest for targeted investigation. In this way, space technology extends its reach downward, guiding expeditions toward features previously unknown. The interplay between ocean and orbit reshapes how the planet is understood. Instruments designed to study climate, monitor sea level rise, and measure polar ice also contribute to geological discovery. A satellite intended to track changes in ocean circulation simultaneously captures clues about underwater volcanoes and plate boundaries. Data collected hundreds of kilometers above Earth reveal structures lying thousands of meters below its surface. Over successive missions, improvements in sensor accuracy and computational modeling refine these maps. Digital reconstructions grow more detailed, narrowing uncertainties and highlighting subtle features. While vast regions of the seafloor remain unmapped at fine resolution, gravity-derived models provide a coherent framework outlining the skeleton of the ocean basins. Through this fusion of spaceborne observation and marine science, the unseen becomes gradually legible. The ocean's surface, once regarded as a barrier to what lies beneath, serves instead as a canvas upon which hidden mass leaves its imprint. By measuring minute variations from orbit, NASA and its partners chart landscapes concealed by depth, demonstrating that exploration of the sea can begin from the vantage point of space. Long before continents settled into their present outlines, before oxygen accumulated in the atmosphere, Earth was a planet shaped by heat and restless chemistry. Volcanoes released gases into a young sky. Oceans interacted with newly formed crust, and energy flowed from the interior as much as from the distant Sunday. In that earlier epoch, conditions near hydrothermal systems may have been common rather than exceptional, providing localized environments where water, minerals, and thermal gradients converged. Modern hydrothermal vents offer a window into that ancient landscape. Within these systems, seawater percolates through fractured rock, becomes heated by underlying magma, and rises again enriched with dissolved metals and reduced compounds. At the boundary where hot fluids encounter cooler surroundings, steep chemical gradients form. These gradients are not merely physical contrasts, they represent stored potential energy capable of driving complex reactions. In mineral-rich environments, certain surfaces provide catalytic properties that influence molecular assembly. Iron-sulfur minerals, abundant in vent settings, play a particularly intriguing role. Their crystalline structures present reactive sites where small molecules can bind and rearrange. Laboratory experiments demonstrate that under appropriate conditions, These minerals facilitate the formation of organic compounds from simpler carbon-bearing precursors. The geometry of their surfaces can stabilize intermediate structures, lowering energetic barriers that would otherwise inhibit synthesis. In such settings, chemistry becomes organized, guided by both thermodynamics and mineral scaffolding. The interplay between water, heat, and rock creates microenvironments within vent systems. Tiny pores and cavities within mineral matrices concentrate reactants, while fluctuating temperatures encourage cycles of reaction and separation. In confined spaces, molecular assemblies can accumulate, increasing the likelihood of complex interactions. Over extended periods, these processes may have contributed to the emergence of self-organizing chemical networks. While no direct record of the earliest replicating systems survives, vent analogs provide experimentally accessible models for testing plausible pathways. Research in this area extends beyond reconstructing the past. By understanding the chemical transitions that may have bridged non-living and living systems, scientists refine criteria for recognizing biological signatures elsewhere. Certain isotopic ratios, specific lipid structures, and patterns of molecular chirality can indicate biological processing. However, distinguishing these from abiotic processes requires careful knowledge of the chemistry that precedes life. Hydrothermal studies help clarify which compounds can arise through purely geochemical means and which suggest metabolic activity. Instruments designed to search for biosignatures on other worlds are calibrated with these distinctions in mind. Spectrometers and chromatographs are tested against samples generated in laboratory simulations of vent chemistry. By comparing organic products formed under controlled abiotic conditions with those produced by living systems, researchers establish baselines for interpretation. The objective is not to assume that life elsewhere mirrors Earth exactly, but to understand the spectrum of chemical possibilities inherent in planetary environments. The investigation of early Earth thus becomes a guide for planetary exploration. If iron-sulfur catalysis and hydrothermal gradients contributed to the origin of life here, similar processes might operate on worlds with liquid water and active interiors. The search for biosignatures then includes attention to mineralogy, redox conditions, and thermal history. Through careful study of modern vent systems and their chemical pathways, a bridge forms between deep time on Earth and distant oceans beyond it, linking questions of origin with the methods used to detect life in unfamiliar settings. For much of modern astronomy, habitability was drawn as a narrow ring around a star. Within that band, temperatures would allow liquid water to persist on a planetary surface, neither frozen solid nor boiled away. Too close, and oceans would evaporate. Too far, and they would harden into ice. The concept was elegant and intuitive, rooted in Earth's own position relative to the Sunday. Distance became destiny, and the search for life focused on worlds orbiting within this circumstellar zone. Over time, that framework began to loosen. Observations of icy moons in the outer solar system revealed bodies far beyond the traditional habitable range that nevertheless showed evidence of internal warmth. Gravitational interactions, radioactive decay, and tidal flexing emerged as alternative energy sources capable of sustaining liquid water beneath thick ice shells. A planet or moon no longer required direct stellar heating at its surface if internal processes could maintain a subsurface ocean. This shift did not arise from speculation alone. It was grounded in the growing understanding of Earth's own deep systems. Beneath kilometers of water and rock, ecosystems thrive without any connection to sunlight. Their energy is drawn from chemical gradients shaped by planetary heat and mineral interactions. These findings demonstrate that life can persist in environments insulated from surface conditions, provided that liquid water and chemical disequilibrium coexist. As telescopes detect exoplanets orbiting distant stars, the diversity of planetary systems becomes apparent. Gas giants migrate inward and outward, rocky planets cluster in unexpected arrangements, and icy bodies may outnumber temperate terrestrial worlds. In such systems, moons subjected to tidal forces could harbor hidden oceans beneath frozen crusts. Even rogue planets, ejected from their parent stars and drifting through interstellar space, might retain subsurface liquid layers if insulated by thick atmospheres or internal heat. The idea of ice covered oceans as common features reshapes expectations. Instead of searching exclusively for Earth like surfaces with blue skies and continents, scientists expand their models to include concealed aquatic environments. These oceans may never interact with starlight, yet they could maintain stable chemical conditions over geological timescales. In this broader view, the habitable zone becomes multidimensional, defined not solely by orbital distance, but by a combination of heat sources, water availability, and geochemical cycling. Deep sea ecosystems on Earth provide the empirical anchor for this redefinition. Their existence demonstrates that biological systems can adapt to pressure, darkness, and isolation, drawing energy from planetary interiors. The criteria for life-friendly conditions shift from surface temperature alone to the presence of sustained energy gradients and solvent stability. The emphasis moves from appearance to process, from visible climates to hidden dynamics. Astrobiology now integrates stellar physics with geophysics, examining how tidal interactions, radiogenic heating, and crustal composition influence potential habitability. Models incorporate interior structure, ice thickness, and ocean chemistry. Remote sensing techniques seek indicators of subsurface liquid layers, such as magnetic field perturbations or plume activity. Each discovery broadens the spectrum of environments considered plausible for life. In this evolving framework, the habitable zone is no longer a simple ring traced around a star. It becomes a network of possibilities shaped by internal and external forces alike. Worlds once dismissed as too cold or too distant enter scientific consideration, not as anomalies but as legitimate candidates. Through insights gained from Earth's own depths, the definition of life-friendly expands, inviting exploration of oceans that lie hidden beneath ice, far from the warmth that once seemed essential. At the margins of chemistry and physics, life persists in forms that seem to press against the edges of possibility. In laboratory cultures and remote field sites, researchers observe microorganisms that tolerate conditions once assumed incompatible with biological structure. These organisms do not merely endure extremes, they incorporate them into stable, metabolic routines, extending the known boundaries of survival. In acidic hot springs and volcanic drainage systems, certain microbes inhabit waters with pH values low enough to dissolve metal. Their cellular membranes maintain internal neutrality through tightly regulated ion transport systems, preventing destructive proton influx. Proteins within these cells are stabilized by specific amino acid compositions that resist denaturation under corrosive conditions. Enzymatic pathways adjust to operate efficiently despite surrounding acidity, allowing metabolism to proceed where most organisms would rapidly fail. Elsewhere, radiation-saturated environments host communities adapted to persistent exposure. Some bacteria possess remarkable DNA repair mechanisms capable of reassembling shattered chromosomes. Redundant genetic copies and specialized enzymes collaborate to correct damage caused by ionizing radiation. Protective pigments and antioxidant systems mitigate oxidative stress, preserving cellular integrity over repeated cycles of exposure. These adaptations demonstrate that genetic information can remain stable even when bombarded by energy levels lethal to more familiar life forms. Pressure presents a different challenge, compressing molecular structures and altering membrane fluidity. In deep marine trenches and subsurface sediments, piezophilic microorganisms adjust the composition of their lipid membranes to remain functional under immense compression. Proteins are structured to retain flexibility despite reduced volume, maintaining catalytic activity at depths where external forces would deform conventional biomolecules. Cellular processes slow, yet they do not cease. Growth may be measured in extended intervals, but replication continues within environments of extraordinary confinement. Temperature extremes add further dimensions. Thermophiles thrive in environments exceeding the boiling point of water at surface pressure, their proteins reinforced by additional ionic bonds and hydrophobic interactions. Psychrophiles persist near freezing, their enzymes remaining flexible and efficient at low kinetic energy levels. Each adaptation represents a solution shaped by natural selection, tuned to specific gradients of heat, chemistry, and radiation. As these limits are documented and quantified, theoretical models of habitability evolve. Parameters once considered fixed become adjustable. If life can endure intense acidity, then acidic oceans on other worlds require reevaluation. If radiation resistance is achievable, then planetary surfaces exposed to stellar flares may not be uniformly sterile. If pressure tolerance extends into deep crustal environments, subsurface oceans beneath thick ice shells become plausible habitats rather than speculative anomalies. NASA integrates these findings into mission design and data interpretation. Instruments are calibrated to detect biosignatures across broader chemical contexts, accounting for organisms that may inhabit unconventional niches. Laboratory simulations incorporate extreme conditions to test the stability of organic compounds and metabolic markers. The search for life expands from a narrow band of temperate environments to a spectrum defined by gradients and resilience. Each newly documented extremophile does not merely add to a catalog of curiosities, it reshapes expectations, informing models that guide exploration beyond Earth. The limits of survival become dynamic rather than static, influenced by empirical evidence drawn from remote deserts, deep sediments, and radiation exposed sites. Through careful study of these organisms, the boundaries of the possible shift incrementally outward, refining the criteria by which distant worlds are assessed for their capacity to host living systems. In laboratories and aboard research vessels, the search for life is translated into chemistry. Samples drawn from water columns, sediments, and vent fluids are filtered, sealed, and analyzed with instruments sensitive to traces measured in parts per billion. Within these measurements, certain molecules carry particular weight. Methane, hydrogen, and a wide array of organic compounds are examined not simply for their presence, but for their context, concentration, and isotopic character. Methane, for example, can arise through biological metabolism or through abiotic reactions between water and rock. Distinguishing between these pathways requires attention to isotopic ratios, especially the relative abundance of carbon-12 and carbon-13. Living systems tend to favor lighter isotopes during enzymatic reactions, leaving behind subtle signatures within the gas they produce. By comparing isotopic distributions with laboratory standards and geological models, researchers evaluate whether methane reflects metabolic activity or purely chemical synthesis. Hydrogen serves as another indicator, often functioning as a potential energy source within microbial ecosystems. Its presence, particularly when associated with specific mineral interactions, can signal environments where redox gradients are active. Scientists measure hydrogen alongside dissolved metals, sulfates, and carbon dioxide, constructing a chemical portrait of energy availability. In terrestrial oceans, such gradients sustain communities that do not rely on sunlight, providing analogs for interpreting measurements from other worlds. Organic molecules encompass a broader category. Amino acids, lipids, and complex hydrocarbons may form through biological processes, yet some also emerge under abiotic conditions. The distinction often lies in patterns rather than isolated compounds. Biological systems frequently produce specific distributions of chain lengths or exhibit consistent molecular chirality, favoring one mirror image over another. Analytical techniques such as gas chromatography and mass spectrometry separate and identify these compounds with high precision, revealing structural details that hint at their origin. Ocean environments serve as testing grounds for these detection strategies. Instruments designed for planetary missions are exposed to challenging conditions underwater, where pressure, temperature variability, and chemical complexity mirror aspects of extraterrestrial settings. Sensors are calibrated against known concentrations of methane and hydrogen, ensuring that sensitivity thresholds align with realistic scenarios. False positives are scrutinized, and interference from background chemistry is characterized in detail. Deep sea expeditions generate datasets that refine algorithms used in space exploration. When instruments aboard spacecraft encounter unexpected readings, researchers compare them to patterns observed in Earth's oceans. This comparative framework strengthens interpretation, reducing ambiguity in environments where direct sampling may be limited. Laboratory simulations extend these comparisons, recreating ocean chemistry under controlled conditions to evaluate how instruments respond to varying mixtures. The process is cumulative and iterative. Each refinement in sensor design or analytical method emerges from empirical evidence gathered in marine settings. By understanding how biosignatures manifest within complex natural systems, scientists sharpen their criteria for recognizing similar patterns elsewhere. The ocean becomes both a source of analogs and a proving ground for technology, linking terrestrial chemistry with distant exploration. In this careful study of molecules and isotopes, the search for life acquires a tangible form. It is not an abstract hope, but a measurable pattern within gases, fluids, and sediments. Through disciplined observation and calibration, the tools carried into space are grounded in knowledge drawn from Earth's own depths, guiding interpretation when signals from other worlds are finally received. Each descent into the deep ocean carries with it the quiet possibility of encountering something never before recorded. Despite decades of exploration, remotely operated vehicles continue to transmit images of organisms that defy classification at first glance. Pale corals arranged in intricate lattices, gelatinous creatures drifting with translucent symmetry, crustaceans bearing unfamiliar appendages, all appear against the dark backdrop of the seafloor. Taxonomists on the surface examine these recordings and specimens with care, often determining that they represent entirely new species, sometimes even new genera. The rate of discovery underscores how incomplete current knowledge remains. Vast regions of abyssal plains, mid-ocean ridges, and trench systems have been visited only briefly, if at all. Sampling efforts are limited by time, funding, and the logistical constraints of deep sea operations. As a result, the catalog of marine biodiversity expands steadily with each expedition. Genetic sequencing further reveals hidden diversity, distinguishing lineages that outwardly appear similar yet diverge significantly at the molecular level. The ocean's depths harbor a complexity that continues to unfold. Bioluminescence emerges as one of the most pervasive traits within this concealed world. Numerous organisms generate light through biochemical reactions involving luciferins and luciferases, producing flashes, glows, and pulses of varying intensity and color. In an environment devoid of sunlight, these emissions serve diverse functions. Some species use light to attract prey, others to signal mates or deter predators. Certain fish possess photophores arranged in species specific patterns, forming luminous signatures that aid in recognition. The chemistry of light production is efficient, converting chemical energy into visible photons with minimal heat loss. Entire communities operate within layers of the water column, seldom sampled. The mesopelagic zone, sometimes referred to as the twilight region, contains immense populations of small fishes and invertebrates that migrate vertically in daily cycles. Below, the bathypelagic and abyssal zones host organisms adapted to stable darkness and high pressure. Many of these habitats remain poorly characterized, and new survey technologies often reveal unexpected assemblages. Hydrothermal fields, methane seeps, and seamounts create localized biodiversity hotspots, each with distinct ecological networks. The challenge extends beyond discovery to understanding ecological relationships. Identifying a new species is only the first step. Researchers seek to determine its role within the food web, its reproductive strategies, and its resilience to environmental change. Long-term monitoring is rare, making it difficult to assess population dynamics across seasons or years. Consequently, scientific interpretation evolves gradually, built from fragments of observation gathered across scattered expeditions. This ongoing revelation carries broader implications. If Earth's largest habitat continues to yield unrecognized forms of life, it suggests caution in assuming completeness elsewhere. Models of habitability and biodiversity must accommodate the possibility of unseen complexity. The ocean demonstrates that even within a well-studied planet, vast biological diversity can remain hidden in inaccessible domains. In this sense, the deep sea functions as both a repository of undiscovered species and a reminder of scientific humility. Its ecosystems persist largely out of view, structured by pressures and chemistries that shape unfamiliar morphologies and behaviors. With each new expedition, the boundaries of known life shift slightly outward, reinforcing the understanding that exploration, whether downward into water or outward into space, proceeds through incremental revelation rather than sudden finality. Silence settles differently when it is layered with depth. Within submerged habitats and sealed research modules, the outside world becomes abstract, reduced to filtered signals and scheduled transmissions. Light arrives artificially, measured and controlled. Beyond reinforced walls lies water pressing steadily inward, a constant reminder of separation. In such environments, the mind registers confinement not as sudden alarm, but as a gradual narrowing of sensory experience. Underwater missions are designed not only to test equipment, but to observe people. Crews inhabit compact living quarters for extended periods, sharing workspaces, sleeping areas, and routines. The absence of open horizons alters perception of time. Days are structured by tasks and communications rather than by sunrise and sunset. Within this contained setting, researchers examine how individuals adapt to limited privacy and repetitive surroundings. Psychologists monitor stress responses through physiological markers such as heart rate variability, cortisol levels, and sleep patterns. Small changes in tone during conversation, shifts in posture, and variations in decision making are recorded and analyzed. These metrics provide insight into how confinement influences cognition and mood. Patterns observed underwater offer a controlled approximation of conditions expected during long duration space travel, where physical isolation is compounded by distance from Earth. Darkness contributes its own dimension. Even when artificial lighting is available, awareness of the surrounding deep persists. The external environment is inaccessible without deliberate preparation, reinforcing a sense of enclosure. This mirrors aspects of interplanetary missions, where astronauts will be surrounded by the vastness of space, separated from planetary surfaces by vacuum rather than water. In both cases, external environments are hostile, and return to ordinary settings requires coordinated effort. Team dynamics become central to endurance. Confined groups must negotiate responsibilities, resolve disagreements, and maintain morale within limited social circles. Underwater analog missions provide opportunities to study leadership styles and communication strategies in real time. Rotating roles, structured debriefings, and conflict resolution protocols are evaluated for effectiveness. Observations from these missions inform training programs for astronauts, emphasizing adaptability and collaborative problem solving. Isolation also shapes cognitive load. Without immediate external input, individuals may experience heightened focus on internal processes. Researchers examine how concentration fluctuates, how creativity is sustained, and how monotony is mitigated through structured schedules. Exercise routines, shared meals, and designated personal time are incorporated to maintain psychological balance. These practices are refined iteratively, informed by empirical data gathered in submerged environments. Human endurance in such settings is not defined solely by physical resilience, but by sustained mental clarity. Decision making under confinement must remain deliberate and precise, especially when communication delays are introduced. Underwater habitats provide a space to observe how individuals manage responsibility when direct oversight is limited. The findings contribute to mission planning for journeys that will extend months or years beyond Earth's immediate reach. In the steady quiet of these submerged stations, lessons accumulate about human adaptability. The interplay between environment and psychology becomes measurable, offering insight into how crews may respond to prolonged separation from familiar landscapes. Through careful study of isolation beneath the surface, preparation for distant voyages gains depth, grounded in observation of how minds and teams function when horizons are replaced by enclosing walls. Deep within fractured rock, water and stone engage in a slow exchange that requires no light. When seawater infiltrates ultramafic rock, rich in olivine and pyroxene, a transformation unfolds at the molecular level. The minerals reorganize, incorporating water into new crystalline structures such as serpentine. In the process known as serpentinization, iron within the rock is oxidized and molecular hydrogen is released as a byproduct. This hydrogen accumulates in pores and fractures, creating pockets of chemical potential within the crust. Hydrogen, though simple in structure, carries energetic significance. In environments where carbon dioxide or other oxidized compounds are present, it can serve as a fuel for metabolism. Microorganisms capable of exploiting this gradient couple the oxidation of hydrogen to the reduction of carbon dioxide, synthesizing organic molecules while releasing energy. The reaction proceeds without any dependence on photons, sustained instead by disequilibrium between reduced and oxidized species. The boundary where these compounds meet becomes a site of biochemical opportunity. In terrestrial settings, serpentinization occurs along mid-ocean ridges and within certain continental regions. Fluids circulating through newly formed crust interact with mantle-derived rocks, generating alkaline solutions enriched in hydrogen and methane. Some vent systems associated with this process, such as those discovered along slow-spreading ridges, host microbial communities that rely heavily on hydrogen metabolism. These organisms inhabit mineral-lined cavities where gradients are sharp and stable over long periods. Chemical gradients form the core of this energy landscape, where hydrogen-rich fluids encounter more oxidized seawater. Differences in electron availability arise. Membranes within microbial cells harness these differences through protein complexes that transfer electrons along chains, generating proton gradients across cellular boundaries. The stored electrochemical potential drives ATP synthesis, enabling growth and replication. In such systems, life is anchored not to sunlight, but to the steady flow of electrons from rock-derived fuels to available acceptors. The implications extend beyond Earth's crust. Icy moons possessing subsurface oceans may contain rocky cores subject to tidal heating. If liquid water interacts with silicate material under sufficient pressure and temperature, serpentinization could proceed in analogous fashion. Hydrogen produced in this manner would accumulate within oceanic reservoirs, potentially providing a continuous energy source. The presence of carbon-bearing compounds and suitable oxidants would complete the basic requirements for hydrogen-based metabolism. Laboratory experiments simulate these conditions by reacting water with crushed ultramafic rock under controlled pressure and temperature. The resulting fluids are analyzed for hydrogen production, pH shifts, and mineral alteration patterns. These experiments refine models of energy availability in subsurface oceans beyond Earth. Instrumentation designed for planetary missions incorporates sensors capable of detecting hydrogen and associated chemical species, informed by data derived from terrestrial analogs. Energy without sunlight thus becomes a tangible framework rather than a speculative abstraction. It rests on measurable geochemical reactions and observed biological pathways. By tracing the interplay between water and rock, scientists identify mechanisms that can sustain metabolism in darkness. In this steady interaction, hydrogen emerges as a bridge between geology and biology, linking planetary interiors to the potential for living systems. Through understanding serpentinization and its consequences, exploration of distant icy oceans gains a grounded foundation rooted in processes already active beneath our own planet's surface. Across the Antarctic plateau, wind moves in long, low currents over a surface that appears unbroken and still. Beneath kilometers of compacted ice, however, liquid water persists in isolated pockets known as subglacial lakes. These reservoirs remain sealed from the atmosphere by immense pressure and cold, insulated by the very ice that conceals them. To reach such environments requires patience, precision, and an approach that preserves what lies below. The parallels to icy moons are clear in structure, if not in scale. On Europa and other ocean-bearing worlds, thick shells of ice likely separate surface conditions from liquid water beneath. Accessing those hidden oceans would demand drilling through frozen layers while preventing contamination from the lander itself. Antarctica provides a terrestrial analog where these engineering challenges can be confronted directly. The ice sheet is thick, stable, and remote, creating conditions that approximate the logistical isolation expected on distant missions. Reaching a subglacial lake begins with mapping. Radar surveys trace the depth of the ice and identify regions where water accumulates beneath. Once a target is selected, drilling systems are assembled with meticulous care. Hot water drilling is often employed, circulating heated, filtered water through boreholes to melt a narrow shaft downward. The drilling fluid is sterilized and monitored, ensuring that microbes from the surface do not descend into pristine environments that have been isolated for millennia. Maintaining sterility throughout the process becomes an engineering discipline. Equipment is cleaned with chemical agents and ultraviolet light. Components that enter the borehole are enclosed and protected during transit across the ice. Tracer compounds are sometimes added to drilling fluids in minute concentrations, allowing scientists to detect any intrusion into collected samples. These safeguards reflect an understanding that once contamination occurs, it cannot easily be reversed. When the drill reaches liquid water, instruments are lowered carefully into the cavity. Cameras, temperature probes, and water samplers collect data without disturbing sediments or introducing foreign material. The conditions encountered within subglacial lakes are extreme, cold, dark, and under significant pressure. Yet microbial life has been detected in some of these environments, demonstrating resilience in isolation. Each successful access provides insight not only into Antarctic ecology, but into the feasibility of penetrating ice on other worlds. Engineers analyze every stage of these operations. They evaluate the stability of boreholes, the durability of seals, and the performance of sensors in near freezing water. Power requirements, communication strategies, and contingency plans are refined with each expedition. The lessons learned extend beyond polar science, informing conceptual designs for probes intended to melt through extraterrestrial ice crusts while carrying sterile instrumentation. The challenge is not solely mechanical. Logistics in Antarctica demand coordination across long supply chains, careful timing within narrow seasonal windows, and adaptation to weather that can shift abruptly. These constraints mirror aspects of planetary exploration where limited launch opportunities and environmental uncertainty require rigorous planning. By confronting such variables in a controlled terrestrial context, mission designers gain experience in balancing ambition with caution. Within the stillness of the Antarctic ice sheet, the act of drilling becomes a rehearsal for reaching concealed oceans elsewhere. Each borehole carved downward represents both a scientific endeavor and a test of responsibility. Through disciplined technique and incremental refinement, the path toward exploring ice-covered worlds grows clearer, grounded in the patient work conducted beneath Earth's own frozen surface. In the deep ocean, instructions cannot always arrive in time. Signals weaken, latency stretches, and the environment shifts without warning. Within that silence, machines must interpret their surroundings independently, adjusting course without waiting for permission. Autonomy becomes less a convenience and more a necessity, woven into the architecture of deep-sea robotics from the beginning. Autonomous underwater vehicles descend without tethers, carrying onboard systems that map terrain, avoid obstacles, and manage energy reserves. Sonar pulses radiate outward, returning echoes that are translated into three-dimensional models of unfamiliar landscapes. Algorithms compare incoming data with internal maps, updating routes in real time. If a current nudges the vehicle off course, inertial systems detect deviation and compensate. When visibility drops or sediment clouds obscure sensors, decision trees determine alternative paths, preserving mission objectives without external intervention. The ocean provides a proving ground for artificial intelligence under constraints similar to those expected in distant planetary exploration. Communication bandwidth is limited, and the cost of transmitting large datasets to the surface can be high. To conserve resources, onboard processors filter information, selecting which measurements merit transmission and which can be archived locally. Pattern recognition software identifies anomalies, flagging features that differ from background geology or chemistry. In this way, the machine becomes not merely a tool, but an analytical partner. Training these systems requires exposure to complex, unpredictable environments. Underwater terrains vary in texture, slope, and composition. Biological communities introduce movement and variability. Engineers feed collected data into machine learning models, refining their ability to distinguish rock from organism, sediment plume from structural feature. Iterative testing sharpens classification accuracy, reducing false positives and improving navigation reliability. Each deployment generates new datasets that enhance the performance of subsequent missions. Power management forms another critical dimension. Deep sea robots operate on finite energy supplies, balancing propulsion, sensing, and computation within constrained budgets. Artificial intelligence assists by optimizing task sequences, shutting down non essential systems when idle, and adjusting sampling frequency according to environmental conditions. These strategies mirror the demands of spacecraft operating far from Earth, where recharging opportunities are rare and every watt must be allocated with care. Autonomy also extends to contingency response. If a thruster malfunctions or a sensor fails, diagnostic algorithms assess system health and reconfigure operations. Redundancy is built into both hardware and software, allowing the vehicle to continue functioning despite partial impairment. Engineers observe these responses underwater, evaluating robustness before similar architectures are integrated into probes destined for icy moons or distant planets. The refinement of AI navigation beneath the waves directly informs mission design beyond Earth. On a world where communication delays span minutes or hours, probes must interpret sensor data, prioritize sampling sites, and adapt to unforeseen obstacles without immediate guidance. The lessons drawn from ocean deployments shape how distant explorers will function when separated from human oversight. Within the abyss, artificial intelligence matures through practical challenge. Each dive becomes a quiet rehearsal for journeys that will unfold across greater distances. By learning to navigate darkness, pressure, and uncertainty in Earth's oceans, autonomous systems gain the resilience required for exploration of hidden seas elsewhere, where independence will define both survival and discovery. Even now, the maps remain incomplete. Vast regions of the seafloor are rendered as softened contours, inferred from distant measurements rather than traced in detail. Beneath those approximations lie valleys, ridges, fractures, and plains that have never been seen directly. The ocean's greatest depths still hold more blank space than clarity, not because of neglect, but because scale and difficulty outpace our tools. Hydrothermal vents were once thought rare, scattered anomalies along mid-ocean ridges. Yet with each expedition, new fields have been located, sometimes separated by hundreds of kilometers from previously known systems. It is reasonable to suspect that entire vent provinces persist beyond the reach of current surveys, hidden along unexplored spreading centers or beneath thick sediment. Some may operate intermittently, flickering on and off as tectonic stresses shift, leaving little surface signal to betray their presence. The chemistry of these environments varies in subtle and significant ways. Differences in underlying rock composition alter the fluids that emerge. Basaltic crust yields one suite of dissolved elements, ultramafic rock another. Temperature gradients shape mineral precipitation. Mixing rates with surrounding seawater influence biological communities. Each vent field becomes a laboratory with its own character, defined by the interplay of geology and fluid flow. Yet we have sampled only a fraction of these chemical combinations. Cold seeps, diffuse flow areas, and subsurface reaction zones add further complexity. Fluids can migrate laterally through porous rock before emerging, carrying chemical signatures that differ from those of focused black smoker chimneys. Microbial ecosystems may flourish in thin films within fractures, far from visible plumes. Accessing such niches requires drilling or carefully positioned sensors, and even then, spatial coverage remains limited. We cannot yet trace every gradient or catalog every metabolic pathway operating in these hidden networks. Beyond vents and seeps, large expanses of abyssal plain remain scarcely visited. Sediments accumulate slowly there, recording biological and chemical signals over long intervals. Microbial communities inhabit these deposits at densities far lower than those found near active systems, yet their cumulative volume may be vast. Determining how these organisms persist, how they exchange energy, and how they respond to environmental shifts remains an ongoing endeavor. Sampling at sufficient resolution across such distances poses logistical and financial challenges. Technological constraints continue to shape exploration. Deep-sea vehicles have finite dive times, limited sensor arrays, and restricted coverage per mission. Satellite altimetry outlines broad topography, but it cannot reveal fine-scale structures or subtle fluid discharge. Even with autonomous systems expanding our reach, coverage accumulates incrementally. Each survey illuminates one region while leaving countless others untouched. The unknown extends to temporal dynamics as well. Vent activity evolves over years and decades. Chemical fluxes change as magmatic systems cool or recharge. Biological communities respond, migrate, or collapse. Continuous monitoring at multiple sites would reveal patterns of succession and resilience, yet such networks are only beginning to form. Our present understanding captures moments rather than full cycles. In acknowledging what remains undiscovered, the ocean does not appear diminished but enlarged. Its complexity grows with each answer, revealing additional layers of structure and variation. The recognition that entire systems may lie beyond our current awareness reflects not failure, but the vastness of the domain itself. Exploration proceeds steadily, guided by evidence and refined tools, while the greater expanse waits with quiet patience beneath the surface. There is a rhythm to returning. The first expedition sketches an outline, the second refines it, and the third begins to notice subtleties that were once invisible. In remote ocean environments, a single visit offers only a momentary glimpse. Processes unfold across years, sometimes across decades, and understanding requires patience measured on the same scale. When research teams revisit hydrothermal systems, they do not simply confirm prior observations. They measure temperature shifts within vent fluids, track changes in mineral deposition, and compare microbial community structures over time. Subtle variations in chemical output can indicate deeper geological adjustments. A slight cooling trend may suggest waning magmatic heat. A surge in certain dissolved compounds can reveal renewed circulation through fractured rock. These patterns are discernible only when data sets accumulate across repeated missions. Long-term monitoring transforms isolated discoveries into narratives of continuity. Fixed instruments placed near vent fields record fluid chemistry, seismic tremors, and biological activity at regular intervals. Cabled observatories transmit data continuously, allowing researchers to detect transient events such as eruptions or collapses that might otherwise go unnoticed. Over extended periods, variability itself becomes a measurable feature, distinguishing stable systems from those prone to rapid change. Biological communities also reveal their character through persistence. Some species colonize new vents quickly, their larvae dispersing through deep currents and settling on fresh mineral surfaces. Others arrive slowly, dependent on precise conditions that may take years to develop. By returning to the same sites, scientists observe succession, migration, and resilience. They learn which organisms anchor ecosystems and which respond most sensitively to environmental fluctuation. Stability emerges not as stillness, but as the capacity to adapt without collapse. These extended observations inform more than oceanography. For space agencies, the value lies in understanding how life behaves when energy sources vary and environmental parameters shift. If a vent field on Earth can sustain communities through episodic disturbances, it suggests that extraterrestrial systems might also exhibit durability. Conversely, identifying thresholds beyond which ecosystems falter refines expectations for habitability elsewhere. Repeated expeditions also strengthen technological capability. Instruments are recalibrated against long-term baselines, improving precision. Sampling techniques evolve as prior limitations become clear. Data management strategies mature, enabling integration of chemical, biological, and geological records into coherent frameworks. The act of returning becomes an iterative refinement of both knowledge and method. Patterns that once seemed random begin to reveal structure. Seasonal influences on deep currents, multi-year cycles in tectonic activity, and gradual shifts in microbial composition become visible only through sustained attention. Decades of observation transform isolated data points into trajectories. The ocean, once perceived as static in its depths, displays dynamics that unfold at tempos slower than human intuition might expect. NASA's continued engagement with these environments reflects recognition that discovery is not confined to first contact. Initial findings capture imagination, but enduring understanding arises from repetition. By committing to long-term study, the agency aligns its exploration philosophy with the temporal scales of natural systems. In doing so, it acknowledges that the most meaningful patterns are those that emerge quietly over time, revealed through steady return rather than singular encounter. Beyond the glow of any star, life would not need to announce itself with forests or oceans reflecting light. It could persist in forms so small and patient that entire worlds might appear barren from a distance. If it exists elsewhere, it may resemble the quiet tenacity of microbes rather than the visible complexity of animals or plants. On Earth, microbial life dominates in numbers and in resilience. It occupies fractures in rock, circulates through heated fluids, and adapts to gradients of chemistry that fluctuate slowly over time. Such organisms require no sunlight, no open sky, no seasons marked by warmth. Instead, they rely on chemical disequilibria, subtle imbalances between molecules that can be harnessed for metabolism. Where hydrogen meets carbon dioxide, where reduced minerals encounter oxidized fluids, energy becomes available in forms invisible to the eye. On icy moons, beneath crusts that may stretch kilometers thick, similar conditions could exist. Internal heating generated by tidal forces or radioactive decay might maintain liquid water in contact with rocky mantles. In those concealed oceans, water interacting with mineral surfaces could generate hydrogen through serpentinization, creating chemical gradients capable of sustaining metabolism. If microbial communities arise in such settings, they would remain shielded from surface radiation and insulated from vacuum, thriving in environments that outwardly appear frozen and inert. The deep ocean on Earth offers an analog for this possibility. In hydrothermal systems and subglacial waters, microorganisms metabolize without photosynthesis, forming ecosystems grounded in chemistry rather than light. Their existence demonstrates that biology can anchor itself to geological processes. The boundary between rock and water becomes a site of potential emergence, where energy flows not from distant stars, but from the planet itself. If life beyond Earth follows similar principles, it may never produce forests or atmospheres rich in oxygen. It may remain microscopic, dispersed within porous rock or suspended in dark waters. Detecting such life would require sensitivity to subtle biosignatures, shifts in gas composition, isotopic ratios that reflect biological processing, or complex organic molecules arranged in patterns unlikely to arise through abiotic chemistry alone. These indicators would not proclaim their origin loudly. They would require careful interpretation and contextual understanding. The expectation of microbial life aligns with evolutionary reasoning. On Earth, single-celled organisms dominated for billions of years before multicellular complexity emerged. Simpler forms require fewer environmental conditions and can tolerate broader ranges of temperature, pressure, and chemical variability. In isolated oceans beneath ice, where energy flux may be limited, microbial ecosystems could persist without ever advancing toward larger organisms. Such possibilities reframe the search for companionship in the universe. Rather than seeking familiar silhouettes or atmospheric signatures visible from afar, exploration may focus on detecting faint chemical imprints. Instruments designed to sense methane anomalies, hydrogen concentrations, or complex carbon chains derive their purpose from lessons learned in Earth's hidden environments. The deep sea, with its reliance on chemical energy and its microbial abundance, becomes a conceptual bridge between our planet and distant worlds. If life exists elsewhere, it may not resemble the landscapes we know. It may reside beneath ice, within rock, sustained by reactions that proceed slowly but persistently. In that quiet persistence, the model provided by Earth's own unseen ecosystems offers guidance, suggesting that vitality does not require sunlight to endure. There was a time when habitability appeared narrow and fragile, a delicate balance of distance, atmosphere, and light. Earth seemed precisely tuned, its oceans tempered by a star at the right remove, its surface sheltered by magnetic fields and layered air. Within that framework, life felt contingent on rare alignment, as though the universe had offered a single, favorable arrangement and no more. Yet the deeper we have looked into our own planet, the more that assumption has softened. Organisms persist where sunlight never reaches, where temperatures fluctuate beyond what once seemed tolerable, where pressure compresses and chemistry shifts in ways that would have been considered hostile. Life does not cling only to temperate shorelines or fertile valleys. It occupies fractures in basalt, circulates within heated fluids, and stabilizes itself along gradients of acidity and salinity that challenge conventional expectations. This expanding awareness alters perspective. Habitability no longer appears confined to a narrow ring around a star, nor dependent solely on surface warmth. Internal heat, chemical energy, and geological interaction become central components of the equation. Worlds that once seemed inert, locked beneath ice, or distant from stellar light acquire new significance. Beneath frozen crusts, or within subsurface oceans, the conditions necessary for metabolism may persist independent of visible sunlight. The shift is not dramatic or abrupt, it is cumulative. Each discovery in extreme terrestrial environments extends the boundary of possibility by a measured degree. Microbes that metabolize sulfur, iron, or hydrogen demonstrate that energy pathways can be diverse. Communities that endure high radiation or crushing pressure reveal biological flexibility grounded in molecular adaptation. These findings do not romanticize harshness, they document capability. As a result, the universe appears less sparsely habitable than once imagined. The criteria for life friendly conditions broaden, not recklessly, but through evidence. Internal heating generated by tidal forces, radiogenic decay, or residual formation energy may sustain liquid reservoirs beneath insulating layers. Chemical disequilibria, arising from rock water interactions, may supply steady sources of metabolic fuel. The absence of direct sunlight no longer equates to the absence of potential vitality. This broader view reshapes not only scientific models but philosophical orientation. Earth remains remarkable, yet it no longer stands as an isolated anomaly defined by fragility. Instead, it becomes one example within a spectrum of environments where life, given time and energy, can establish persistence. The deep sea, once perceived as peripheral to the narrative of habitability, emerges as central evidence that resilience is intrinsic to biology. In recognizing adaptability, the universe feels less distant. Planets and moons, once dismissed as sterile, are reconsidered with cautious openness. Exploration strategies evolve accordingly, guided by the understanding that life may inhabit niches far removed from familiar climates. Instruments are designed to detect subtle chemical imprints rather than visible greenery, acknowledging that survival may unfold quietly and invisibly. The larger perspective does not diminish wonder, it refines it. Instead of resting on a single extraordinary configuration, life appears as a process capable of integrating with varied physical contexts. Earth's extremes illuminate that capacity, revealing a pattern of endurance woven into the fabric of biology. Through that lens, the cosmos seems not necessarily crowded, but plausibly receptive, its possibilities expanded by the lessons drawn from our own planet's hidden depths. It is a quiet realization, one that arrives gradually rather than all at once. The vast ocean beneath our own planet does not simply host unusual creatures or obscure chemical systems. It reflects conditions that echo across distant worlds. In its darkness and pressure, in its reliance on internal energy rather than sunlight, it becomes a familiar analog for places we have never touched. Beneath kilometers of water, entire ecosystems persist without reference to the sky. Their energy arises from geological processes, from interactions between rock and fluid that generate chemical gradients sufficient to sustain metabolism. These environments are not hypothetical constructs, they are measured, sampled, revisited. The realization that similar interactions could occur beneath icy crusts elsewhere reframes the search for life beyond Earth. The extraordinary becomes continuous with the ordinary, linked by shared physical principles. The ocean's hidden realms remind us that absence of visibility does not imply absence of activity. From the surface, the deep appears uniform and silent. Yet, instruments lowered into that darkness reveal complexity layered upon complexity. Microbial networks weave through sediments and fractures. Chemical fluxes pulse through hydrothermal systems. Biological communities assemble around energy sources that would have been overlooked by earlier generations of science. The unseen becomes structured and dynamic. once attention is directed toward it. In contemplating distant oceans sealed beneath ice, the analogy is no longer abstract. We understand, through direct observation at home, that liquid water in contact with reactive rock can generate persistent energy flows. We have documented organisms that metabolize hydrogen, sulfur, and methane. We have observed stability emerging from what once seemed inhospitable. These findings do not guarantee life elsewhere, but they remove the presumption that sunlight is the sole gateway to vitality. The emotional shift is subtle. Rather than imagining alien biospheres as wholly separate from terrestrial experience, we recognize continuity. The same physical laws govern both. Chemistry proceeds according to shared constraints. Thermodynamics applies without preference for location. If conditions align, even in concealed oceans orbiting distant planets, the processes that sustain life on Earth could unfold there as well. The deep sea becomes less an exotic outlier and more a template. This awareness narrows psychological distance. The unknown is not confined to remote galaxies. It resides within our own planet, in volumes we are still learning to explore. Each descent into darkness carries implications that extend outward. Each new species, documented in extreme conditions, reinforces the notion that biology is adaptable in ways once unimagined. The search for alien life begins not with speculation, but with disciplined observation of what already exists. Standing at the shoreline or observing satellite images of icy moons, one can sense the continuity without dramatization. The ocean beneath us mirrors distant possibilities through shared mechanisms rather than shared appearances. By studying our planet's hidden systems, we refine the lens through which we view the universe. The unfamiliar grows less distant, connected by threads of chemistry and persistence that stretch from seafloor to star. Before rockets rise and trajectories are calculated, there is another descent that takes place. Instruments are lowered through cold water, hulls are sealed against pressure, and researchers turn their attention toward environments that test the boundaries of biology. In studying the deep ocean, NASA is not shifting its gaze away from space, but refining it through proximity. At its greatest depths, Earth presents conditions that approach the operational and biological limits of endurance. Temperatures fluctuate near freezing or surge near hydrothermal outflows. Pressure compresses structures and challenges materials. Light vanishes entirely. In these settings, life persists through metabolic pathways once considered improbable. By examining how organisms function here, scientists gain insight into the thresholds that define survivability. The deep ocean becomes a laboratory where the margins of life are mapped with care. This knowledge shapes the search beyond Earth. Instruments intended to detect subtle chemical signatures must be calibrated against complex natural backgrounds. Sensors are tested in environments rich with methane, hydrogen, and dissolved minerals, ensuring that they can distinguish biological patterns from purely geological ones. Field trials refine sampling protocols, data processing algorithms, and contamination controls. Each adjustment strengthens confidence that when a probe encounters unfamiliar chemistry elsewhere, its interpretations will be grounded in experience. Mission design evolves in parallel. Vehicles are engineered to operate autonomously in darkness, to manage limited energy reserves, and to endure prolonged isolation from direct oversight. Underwater expeditions provide opportunities to evaluate navigation systems, communication strategies, and fault tolerance in conditions that mirror the uncertainty of distant worlds. The ocean offers resistance, variability, and constraint, all essential elements in preparing hardware for environments where intervention is impossible. Human preparation unfolds alongside technological development. Extended deployments in submerged habitats simulate confinement and delayed communication. Crews adapt to restricted space, routine maintenance tasks, and reliance on internal systems for safety. These experiences inform training protocols and psychological research, clarifying how individuals and teams respond to sustained separation from the surface. The lessons learned are not abstract, they are embodied in procedures that will accompany future explorers. Underlying these efforts is a question that has endured across centuries: whether life is an isolated phenomenon or a recurring pattern in the universe. The deep ocean does not provide a direct answer, but it expands the context in which the question is asked. By revealing that life can adapt to extremes of pressure, chemistry, and darkness, it challenges assumptions about rarity. It suggests that habitability may emerge wherever energy gradients and liquid media coexist, even if conditions appear austere. The ocean thus serves as rehearsal space for inquiry itself. It allows hypotheses to be tested against tangible systems, permits instruments to be refined before departure, and grounds speculation in measured reality. Exploration downward informs exploration outward, linking the seafloor to the stars through shared principles of physics and biology. The search continues in both directions. Satellites and probes extend our reach beyond the atmosphere, while submersibles and drills descend beneath the surface. Between these trajectories lies a continuity of purpose, guided by observation and sustained by patience. In studying the depths of our own world, we prepare to recognize life, should it exist elsewhere, not as an anomaly, but as a possibility woven into the fabric of the cosmos.